So for our talk today, it's my great pleasure to introduce our speaker, Dr. Robert Vasser from the Feinberg School of Medicine at Northwestern University. I think it's fair at this point that we can call Robert a legend in the field of Alzheimer's research due to his numerous and notable accomplishments. Among his many accomplishments is the identification of BACE1 as the major beta secretase enzyme and the creation of the 5x FAD mouse model, which I'm sure we've all used at some point. Dr. Vasser received his PhD in 1992 in molecular genetics and cell biology in the lab of Elaine Fuchs at University of Chicago. He then did his postdoctoral fellowship in the lab of Dr. Richard Axel at Columbia University, where he investigated the molecular neurobiology of olfaction. After his postdoc, he joined the biotechnology company Amgen to start a research group to identify novel drug targets for AD, which is where he made the BACE1 discovery. In 2001, Dr. Vasser moved to Northwestern University Feinberg School of Medicine, where he continues to explore the molecular and cellular mechanisms of AD. Robert has had continuous NIH funding and has won numerous prestigious awards, including the Potemkin Prize for Alzheimer's Research in 2009, the Alzheimer's Association Zenith Fellow Award in 2012, and the Davey Professor of Alzheimer's Research Endowed Chair in 2019. And with that, I'll turn it over to Robert.
Thank you, Brent. That was really a lovely introduction. So we lost a dear friend and colleague on March 12th, Steve Wagner, who, as you know, was in the Department of Neuroscience at UCSD. Steve was a leader in the field of gamma secretase modulators. And he and Dr. Kevin Ryerson were very close to an investigational new drug application in a phase 1 clinical trial before Steve's passing. Kevin, I understand, is going to continue moving the gamma secretase modulator project forward into the clinic, and we wish him the very best of luck in keeping Steve's legacy alive. Steve was a kind, generous, and loyal friend to all of us who knew him. I'm dedicating my talk to him today in his memory, and in fact, I would not be giving you this seminar today if it were not for Steve, who connected me with Brent. It was last November, I received an email addressed to me and CC'd to Brent from Steve with the subject line, the VAS. The message simply read, one of my favorite AD researchers, and if he has an hour for a Zoom, he would do it for us. And my reply back to Steve was, anytime, Steve, just say when, the VAS. P.S. You are one of my favorite AD researchers too, brother. Steve has left us all too soon, and he will be dearly missed. So this is in honor of you, Steve. May your spirit and legacy live on. So what I want to do today in my talk is to give you some work on some relatively describe a project that is relatively new in our lab. I'm known for BACE, but instead I'm going to talk to you about ACE. Just take the first letter off of BACE and there you have ACE. And it turns out that angiotensin converting enzyme is a risk factor for Alzheimer's disease. And I'll go into detail about characterization of a mutation that we co-discovered with Rudy Tanzi and characterized, and hopefully convince you that it has a role in Alzheimer's disease. So first, I'll give you a background into angiotensin converting enzyme and its links to Alzheimer's disease. Then I'll describe the mutation that Rudy's team identified that has that infers an increased risk for Alzheimer's disease, the R1279Q mutation. And then I'll describe very briefly, if I have time, some future directions and therapeutic implications for Alzheimer's disease. So this work was performed by a very talented research assistant professor in my group, Dr. Leah Cuddy, and shown here on the left. This was published in Science Translational Medicine. As you all know, I don't really need to give this group much of an introduction to Alzheimer's disease. There's severe brain atrophy. Neurodegeneration and neuron death is a hallmark of the disease, as well as the plaques and the tangles and inflammation. There are as you know, there are many unsolved mysteries of Alzheimer's disease, but I think three of the top ones on my list are age is a primary risk factor for AD, selective neuronal vulnerability. Why do some regions of the brain degenerate, like in the hippocampus, yet others in the cerebellum are relatively spared? And why do women have a higher risk for Alzheimer's disease than men? And I think our work in the ACE variant that I'm about to describe, I think will provide us some insight into those questions. This is a figure that Dave Holzman put together a while ago, showing how the amyloid plaques and the neurofibrillary tangles are related to the different cells throughout the brain. I don't think I need to go into a lot of detail here, except that amyloid pathology precedes tau pathology by perhaps decades in the human brain. And the amyloid pathology is thought to be a trigger for the neurofibrillary tangle formation and the ultimate neurodegeneration in the brain. And that there are multiple genetic risk factors involved in Alzheimer's disease. There are the very few autosomal dominant mutations in amyloid precursor protein and presenilin 1 and presenilin 2. Of course, there's the ApoE4 genotype that predisposes to Alzheimer's disease. Other famous genes of late are TREM2, which is expressed in microglia. Microglia have, and the immune cells of the brain are the innate immune cells of the brain, and they have a large role in the pathogenesis of Alzheimer's disease. There are also a huge number of more common variants that have a low risk for Alzheimer's disease. Many of these are expressed in microglia. And we have yet to really decipher how all these different genes interplay with one another to increase the risk of AD, although we do know a lot about the autosomal dominant mutations at this point, and variants in the protein coding regions of these genes provide the most information, the most biological information on the mechanism of AD pathogenesis. So this is a recent GWAS from Kunkel et al. Showing the Manhattan plot of all the genes, risk factor genes that are involved in Alzheimer's disease. And there were 5 new ones that were mentioned in this paper. One of them was ACE, angiotensin converting enzyme, and it just made the significance, the genome-wide significance threshold of 5 times 10 to the minus 8. And here's the p-value right here. So at the time of this study, ACE was not a validated risk factor gene. When this study came out, we were right in the middle of our work on the ACE R1279Q variant that Rudy discovered. And so we were really pleased by seeing this result that ACE finally became a validated AD risk factor gene. Here's angiotensin converting enzyme here shown in a molecular model on the right. ACE, now there are 2 ACEs. ACE1 is the one shown here. It's involved in blood pressure control by cleaving angiotensin 1 into angiotensin 2. It has a number of other substrates, including A beta itself, which is interesting. There's a second ACE, ACE2, which is, as you now know, probably is the receptor for the SARS-CoV-2 virus for its entry into cells. It does have a role in blood pressure control, but as far as we know, it does not have a role in Alzheimer's disease as of yet. So this talk is going to be focusing mainly, exclusively actually, on ACE1. So ACE1 is expressed at significant levels in all tissues, including the brain. But no one really understands the function of ACE1 in the brain, but it's expressed at different levels in different neuronal subtypes. For example, cerebellar neurons express high levels, relatively high levels of ACE1, whereas Neurons in the hippocampus express relatively low levels of ACE. It's a type 1 membrane zinc-dependent dipeptidase with two homologous catalytic domains shown here. And both of these catalytic domains are active and they do cleave substrate. And they have different substrate specificities. So it's quite a complicated protease. And to this day, we don't fully understand what ACE is doing. It has multiple functions. Obviously, one function is in blood pressure control, but it's expressed in neurons in the brain and in a wide variety of neurons. And we still don't know what it's doing in the brain, in neurons specifically. ACE is membrane bound and it's shed from the cell surface by an atom family protease, releasing the catalytic domains into the circulation for blood pressure control. And likely it's also released into the interstitial fluid of the brain to float around and cleave substrate. And again, we don't understand its function in that regard. There is a lot known about the renin-angiotensin system in the periphery, in which ACE is a central player. And this is just the RAS system shown here. And angiotensinogen is cleaved by renin into angiotensin 1. ACE1 then cleaves angiotensin 1 into angiotensin 2, where it then acts on angiotensin receptors, specifically the AT1 receptor, which has multiple functions in different tissues. So this is a very complex pathway involving many tissues of the body. And it's also, ACE happens to also be involved in the peripheral immune response. It modulates neutrophil and macrophage function, and it's highly expressed in monocytes upon differentiation into macrophages and dendritic cells. So this might give us a clue as to what it might be doing in the brain. And we haven't, one area of future study for us is to explore the function of ACE in activated microglia. So it we haven't verified this, but it's highly possible, in fact, probable, that ACE1 is upregulated in activated microglia. So there are a number of past hints that connect ACE1 with Alzheimer's disease. For a long time, it's been known that midlife hypertension has been associated with an increased risk of Alzheimer's disease. Also, epidemiological studies have shown that either ACE inhibitors or angiotensin receptor blockers that are prescribed for reducing hypertension are actually reduce the risk of Alzheimer's disease. But these are epidemiological studies and correlative, and so they don't really, give us a real proof of that particular connection. There's a common ACE gene insertion deletion. It's an ALU indel into intron 16, and it's associated with different ACE1 levels. This ALU sequence, the insertion increases AD risk, but it also reduces hippocampal volume and decreases ACE1 serum levels, especially in women. So, as I said earlier, the risk of Alzheimer's disease in women is greater than in men. And so this might be one connection with, or one clue as to why women might have a higher risk. There are elevated levels of ACE consistently observed in postmortem AD brains. ACE1 degrades A beta in vitro, and there have been a number of studies showing this. For example, the Selco group quite a while ago, but the A beta degrading effects of ACE in vivo have been inconsistent. So some show that in ACE transgenics, that there's decreased levels of A beta in the brain. Other transgenic studies show that there's no difference. So it's not really clear what effect ACE1 may be having on the A beta economy in the brain. And finally, I've already told you about this GWAS paper that came out. Several GWAS papers actually came out implicating ACE as a validated AD risk factor gene. So this all started with Rudy Tanzi and Dmitry Prokopenko at MGH. And so, as you know, Rudy's a geneticist and performing whole genome sequencing and whole exome sequencing. He found families with rare AD-associated variants in the protein coding regions of ACE1. And one of these was R1279Q. The R79, so they screened 446 AD families, and this revealed several rare coding variants in the ACE1 gene. The R1279Q variant was present in 10 families with 13 affected carriers, 2 of which had a mother and child relationship, and there were only 2 unaffected carriers that were not positive for disease. They were about 70 years old, right at what appears to be the age of onset for this variant at the age of 70. So it's possible that these 2 unaffected carriers may have actually gotten Alzheimer's disease at a later age. So this analysis led to us choosing R1279Q for further analysis because of its strong association with AD families. We made knock-in mice using CRISPR-Cas9 genome editing. And this was in the mouse endogenous ACE1 gene. And because there are a few additional amino acids in the mouse ACE1 gene, the cognate variant, the cognate mutation would be R1284Q. And so I'll be referring to R1284Q in the knock-in mouse studies, but it's exactly in the cognate position for R1279Q in humans. These mice were made on a black 6 background. And just to tell you a little bit more about this mutation, it's located at the C terminus of the transmembrane domain of ACE1. And as you probably know from a large number of type 1 transmembrane proteins like APP, APLP2, Notch, ERB4, E-cadherin, there typically are strings of lysines or arginines right at the C terminus of the transmembrane region. And this adds positive charge to anchor the type 1 transmembrane protein into the membrane. And shown down here in this sequence this is the C terminus of mouse and human ACE, and the blue is the transmembrane region. The red R is the site of the variation where Q replaces R in the variant, and then the green is just the C-terminal tail. You can see that there are not very many lysines and arginines in this region. If you take, in fact, this arginine at R1279 is really about the only one. There's another one, a few amino acids further down, C-terminal, but there really there's not a lot of consistent positive charge at the end of this transmembrane region. And so you're taking out, one of the only positive charges at the end of this transmembrane domain. So our hypothesis is that this is somehow destabilizing or changing the position of the transmembrane domain in the membrane, and that this might have an important effect on the function of the enzyme. Like shedding, for example, if it affects the shedding, then there would be less release or more release of the catalytic domains into the interstitial fluid. And again, this is the molecular model of ACE. The R1279, 1284Q mutation is right here at the very C terminus of the transmembrane domain. So it's a very funny place to have a variant that might be affecting the function of an enzyme. I liken it to the tail wagging the dog. And but I think that there is something to this idea that the stabilization in the membrane might be holding a key to the role of this variant in Alzheimer's disease. So what Leah ended up doing is taking primary neurons from the cortex of these knock-in mice that we made. And she did some immunoblots, both cell surface and intracellular immunoblots to look at the levels of ACE. And she found consistently that in the knock-in mice, homozygous knock-ins, there's a higher level of cell surface ACE by using biotinylation assays. And then when she, just looked at the lysate versus the conditioned media, she saw that the lysates have about twice as much ACE in the cell lysates and almost half of ACE in the media, suggesting that there was reduced shedding that was caused by this variant. She then went on to do some primary cell culture neuron experiments like a tunnel assay looking at viability. And she saw that there was both at 7 and 14 days in cultures, the homozygous knock-in neurons exhibited much greater tunnel labeling than wild-type neurons. There was more cleaved caspase 3 by Western blot, and also increased levels of phospho H2AX, which is a marker of DNA damage. So these knock-in neurons were not as healthy in culture. They definitely were exhibiting higher degrees of neurodegeneration just spontaneously. Now, one of the first things we did was measure the blood pressure in these mice because of all the history of ACE1 involvement in blood pressure control. But we found no differences in blood pressure in these mice in either and independent of aging. So whether we looked at them at 8 months or 14 months, the knock-ins did not have increased systolic or diastolic blood pressure. We also did some MRI imaging in our small animal MRI facility at Northwestern and found that there was no difference in cerebral blood flow or blood-brain barrier permeability in the knock-in mice. So this would infer that central rather than peripheral effects of the ACE1 variant are likely responsible for any brain phenotypes observed in the knock-in mice. So this is not a perfect proof of that, but I'm going to suggest that in the following slides, when I show you the phenotypes, that these phenotypes are related to the function of ACE in the brain and not anything to do with the cerebrovascular functions or peripheral functions of ACE. So Leah went on to then look at the brains of these knock-in mice and found that, first of all, there were higher levels of ACE1, just as she saw in the primary neurons in culture. There's higher levels of ACE1 in neurons in vivo. These are cortical neurons here. These are neurons in the cerebellum. The neurons just have higher levels of ACE1. And neurons are the primary source of ACE1 in the brain. We co-stained with IBA1 for microglia and GFAP for astrocytes, and we found very little evidence of ACE1 expression in microglia and astrocytes. Now, these are homeostatic microglia and astrocytes, and so we haven't done a thorough analysis of activation. I wouldn't doubt that ACE1 goes up upon activation. We also saw that there are different levels of ACE1 in different regions of the brain. Now, this is true for wild type mice, shown in the first 3 lanes, but these levels, irrespective of the brain region of ACE, are increased in the homozygous knock-in mice. Cerebellum expresses the highest amounts of ACE in the brain, and cortex an intermediate level and hippocampus a lower level. The other components of the renin-angiotensin system, like angiotensinogen, renin, and AT1R receptor, sort of are interesting. Well, angiotensinogen doesn't really change, but what's interesting about renin and angiotensin 1 receptor are that they have an inverse relationship with the amount of ACE1. For example, in the cerebellum, which has got a lot of ACE, there are conversely the lower levels of renin and angiotensin receptor. So what I think might be going on is that there's a type of homeostatic control of renin angiotensin system signaling in the brain, such that regions that have high ACE may have low renin and receptor to counterbalance that high level of ACE. So Leah went on to look at aging. How was aging affecting these knock-in mice? And one of the first things she saw was that age alone, even in wild-type mice, increases the levels of ACE1 in the brain, shown here by Western blot. Now, when you look at the knock-ins, they already start out at 3 months with higher levels of ACE, and the levels of ACE increase even further at 12 months of age. So there's a knock-in dependent increase of ACE, but there's also an age-dependent increase of ACE that's independent of this variant. Angiotensin 2 levels in the knock-in mice are increased, and I believe this is at 12 months of age, as is signaling through the AT1 receptor, which is mediated via phosphorylated ERK, which is shown here. Now, when Leah started looking at the brains themselves, at 8 months of age, shown here on top, with new end staining, there really wasn't a big effect of the knock-in at 8 months of age, even in the homozygous knock-in shown here. But at 14 months of age, you could see a gene dose dependent effect on brain atrophy, so that the homozygous knock-ins have quite dramatically increased ventricular volume, shown here quantitatively, and also reduced hippocampal volume, a nice gene dose dependent effect in the hippocampus, and that's total hippocampus, and this is dentate gyrus volume specifically. So the R1284Q variant causes hippocampal atrophy and a gene dose dependent effect, and appears to be autosomal dominant. When we started looking in closer into neurodegeneration in the hippocampus, which seemed to be particularly affected by this variant, we saw that specifically in CA1 and CA2, there was increased neurodegeneration in the knock-in mice. So that's shown here at low power, and then when we zoom in at higher power with new end staining, you can see that number of new end neurons is significantly decreased. And it may be a little bit hard to see here, but these arrows are pointing at pycnotic nuclei in the CA2 region that indicates apoptosis. And so, this loss, this neurodegeneration is quantified down here. This is new end covered area of different brain regions, either total brain, which is a slight non-significant trend. Cerebellum shows no difference in the knock-in mice. Hippocampus is a significant decrease. And cortex really doesn't show much of a change either. So there's a selective loss of hippocampal neurons in these ACE1 R1284Q mice. Other brain regions appear to be more resistant to the neurodegeneration. And this is also true for neuroinflammation. There is increased neuroinflammation markers as well as decreased presynaptic markers. And this is actually occurring more severely in females than in males. Although males do show an effect, the females do seem to show a more severe effect. So there's, by Western blot, there is an increase in the knock-in mice for GFAP and for IBA1, indicating neuroinflammation. There's also a decrease in synaptophysin levels. And that's shown here. You can see that, and these were matched fluorescence images shown here. You can see that in wild type, there's significant synaptophysin labeling in the hippocampus, but this is diminished in the knock-in mice. We looked at behavior and electrophysiology using EEG recordings. I'm only showing you the behavioral analysis here. There were deficits in, particularly in the female mice. The male mice did not show, they showed a trend, but not significant impairment in the Morris water maze or in fear conditioning. This is time to platform in the female mice is reduced, as well as the number of platform crossings and quadrant times are affected in the females. Females are particularly affected in fear conditioning, both cued and contextual fear conditioning. So there are behavioral changes. There are abnormalities in the EEG traces of the mice. Again, it's more severe in the females rather than the males. Now, one experiment that was really important that Leah did was to see if she could rescue the neurodegeneration with an ACE1 inhibitor and an angiotensin receptor blocker that are commonly used for the treatment of hypertension. So she chose two drugs that are relatively permeant across the blood-brain barrier, the ACE inhibitor captopril and the angiotensin receptor losartan. And so she treated these mice starting at the age of 9 months of age, and then gave them 6 months of treatment. So then she sacked the mice at 15 months and then analyzed the brains shown here. And as you can see, if you look at this high power magnification here, so vehicle treated mice, the knock-ins have severe neurodegeneration in the hippocampus. This is in the CA1, CA2 region. Captopril rescues this effect as well as losartan. So, and that's quantified down here. So here's the reduction in hippocampus volume in the vehicle, rescue by captopril and losartan. And this is the dentate gyrus decrease of the vehicle and then rescue again. Also, new and covered area is rescued by captopril and losartan. So this was a very important result showing that these drugs that are commonly used for hypertension do rescue the neurodegeneration effect. And it does really, in my mind, prove that this neurodegeneration is occurring through overproduction of angiotensin 2 and overstimulation of the AT1R receptor, which in other models, for example, Parkinson's models of mice, have been linked to neurodegeneration. So that was quite an important result. And then finally, what she wanted to determine was whether crossing the R1284Q mice into an amyloid model what effect that would have. What effect would amyloid have on the effects of the ACE variant? So she crossed the ACE knock-in mice with 5X FAD mice. And then she analyzed the mice at 6 months of age. This is a 5X FAD mouse on an ACE1 wild type background. You can see all the amyloid staining in green, the plaques in green, and nu-N in red. This is an age match 5X FAD with a homozygous knock-in background. And you can see already there's very, very large ventricles, and shown here also is decreased hippocampal volume and overall volume, and then dentate gyrus volume. There's a trend in the heterozygous knock-in mice, but it's not significant. So there's a hint of a gene dose dependent effect, but not, not significant. So the AD pathology reduces the age of onset of neurodegeneration from which occurred at 14 months of age in the knock-in alone, and reduced it down to 6 months of age when on an amyloid background. So there was also when we crossed the knock-in mice to the 5X FAD mice, we saw an even further exacerbation of neuroinflammation. This is 5X FAD on a wild type background versus 5X FAD on a knock-in background. So there's increased IBA1, increased GFAP. Although interestingly, A beta 42 levels were not affected by the ACE1 knock-in when crossed to 5X FAD mice. So in my mind, this kind of goes along with the some of the studies that showed that ACE transgenic mice did not show an effect on amyloid pathology. And so ACE1 really might not be playing a big role on degrading A beta in the brain, but it's doing something else, probably increasing angiotensin 2 to toxic levels and causing neurodegeneration. Again, here's immunostaining here to show increased, at least in the case of IBA1, that there's increased IBA1 here. It's hard to see GFAP. And this is so we wanted to make sure that we're not just following some sort of weird mouse phenotype. We wanted to make sure that this applies to human AD brain. So we went ahead and got AD brains from the Northwestern Alzheimer's Disease Research Center, as well as the Rush Alzheimer's Disease Research Center, and did some Western blotting and some immunohistochemistry. And first, by Western blot, it's really remarkable that AD brain has about over a twofold increase in the level of ACE1 in postmortem human brain. Now, this is frontal cortex. Now, the angiotensin receptor has a trend for a decrease, but it's not significant. And this is compared, obviously, to control, non-demented control brains. The important point, I think, is that when you look at the relationship between ACE1 level and angiotensin 1 receptor in normal control brain, which is shown here by the black dots and the black line, there is a negative relationship between the receptor and ACE1. So the more ACE1 you have, the lower the level of receptor. And I think that then again speaks to this idea I mentioned earlier about some sort of homeostatic regulation of renin-angiotensin signaling in the brain. But this correlation is completely lost in the human AD brain when you look, and that's shown here by the red squares and the red line. So this homeostatic relationship, if it is one, is lost. And this is just staining of a normal non-demented brain with ACE1 and NUN, showing that neurons in the normal brain are the mostly the cell type that expresses the most ACE1 in the brain. Now, in human AD, there's a lot of neuron loss here, yet there's still, I'm sorry, there's neuron loss, but there's some loss in, or at least shift of ACE1, perhaps away from neurons. And so this is where I'm wondering whether these are microglial cells. And we have yet to do that experiment, but that's on our list to do in the future. So I just want to sum up in the last few minutes with our working hypothesis here. And so I think it was important for us to do the human AD experiments to show that there is a dysregulation of ACE, even in regular old garden variety sporadic Alzheimer's disease. And these are people that do not have this very rare R1279Q variant in ACE. So I liken it to, the effect of in cholesterol. If you guys remember your early, probably you learned this in college. So the Nobel Prize was given to Brown and Goldstein for discovering mutations in the LDL receptor that prevent the LDL receptor from internalizing cholesterol into the liver cells and therefore lowering serum cholesterol levels. These people have familial hypercholesterolemia. And these mutations in LDL receptor are extremely rare. But that discovery by Brown and Goldstein, and it led them further, as well as other groups, to explore the cholesterol pathway that led to the identification of HMG-CoA reductase as a rate-limiting enzyme of cholesterol synthesis, which is the target of the statin drugs that we almost all of us now use for lowering serum cholesterol. And so, very rare mutations in coding regions of genes can give us an insight into very important biological pathways for disease. So, I think our ACE variant right here is implying to us that there is a story here that is of importance for even garden variety Alzheimer's disease. So this is our working hypothesis. The R1279Q variant, as I showed you, has increased cell surface level and both cell surface levels as well as total neuronal cell levels. And this leads to an increase in activity just by mass action and the production of more angiotensin II. Now, we're hypothesizing that there's a toxic level of angiotensin II, which then causes hippocampal neurodegeneration. This could be either through a cell autonomous or a non-autonomous pathway. In the autonomous pathway, the hippocampal neurons, which express low levels of ACE, but have high levels of receptor, would be making too much, by having the variant, they would be boosting their level of ACE and making too much angiotensin II, and therefore committing suicide, essentially, if that angiotensin II reaches a toxic level, which could activate NADPH and oxidative stress capsase activation and neuronal death. In the non-autonomous pathway, which is in some ways more interesting, I think, there could be upstream neurons, for example, neurons in the brainstem, certainly cerebellar neurons, that express higher levels of ACE, but may have lower levels of receptors. So they would be more or less immune to the effects of overproduction of ACE and angiotensin 2. But if they synapse upon sensitive neurons, like neurons in the hippocampus, and then secrete angiotensin 2 at toxic levels onto these hippocampal neurons, and perhaps there could be neurodegeneration in this non-cell autonomous pathway. And so I think that this variant provides some insights into the role of aging in Alzheimer's disease. As I showed you, angiotensin 1 increases with age normally in normal wild-type mice. We don't know whether that is occurring in humans, but we suggest that it might be. So it is a potential connection of ACE1 and aging with AD. It also provides insight, I think, into selective neuronal vulnerability, since the level of AT1 receptor is basically going to dictate which neurons of the brain are going to be vulnerable or resistant. And we all have a lot of interest these days into why there are individuals that have resilience or resistance to the pathologies of the brain, the A beta and tau pathologies. And as I also mentioned, the female knock-in mice had more of a susceptibility to neuroinflammation, to deficits in the water maze and in fear conditioning. So it also may give us a clue to female susceptibility to Alzheimer's disease. And then A beta and tau and age itself might be second hits that then exacerbate the level of the effect of the R1279Q mutation. Or in the case of people that don't have R1279Q, that age itself is increasing ACE1 levels normally. And then if it gets hit with a second strike, a second lesion like A beta or tau pathology, that might lead to neurotoxic levels of angiotensin 2. All this suggests that the angiotensin receptor blockers or ACE inhibitors may be useful as a therapeutic for the prevention of Alzheimer's disease. I don't think that they're going to help people that already have AD, but it would be interesting. You know, the epidemiological studies already suggest this, that the ACE inhibitors and the ARBs are protective for Alzheimer's disease. So I think we ought to be investing more effort into understanding that. I'm going to skip the summary, other than to say we should think about repurposing these brain penetrant ACE inhibitors and angiotensin receptor blockers to protect against AD. For future directions, we really need to understand more about the molecular mechanism of our 1279Q. Is it really this altered stability of the transmembrane domain in the membrane that is involving shedding? Or is it increasing half-life or somehow weirdly altering enzyme kinetics? It's not in a position to do that. It's not at a position to do that, but we can't exclude it at this time. So we also want to understand the normal function of ACE1 in the brain. We have some ACE conditional knockout mice where we knocked out ACE in neurons, forebrain neurons, that we're now investigating. And it's a little too early to tell you about those, but we're hoping to gain insights into the normal function of ACE, since it's really such a big mystery. What is ACE doing being expressed by neurons in such a wide regions of the brain? We also need, if we want to move forward with the ACE inhibitors and the ARBs for the prevention of AD, we need to determine which ones are going to be the best to rescue neurodegeneration. And For example, telmisartan is an ARB with, as far as I can tell, the best blood-brain barrier penetration. So we have an experiment ongoing in these knock-in mice with telmisartan. So this is the group here. Leah, shown right here, she's the person who did the all of the analysis with help from her technician, Yelena, right here, and May. And I also want to mention Eric Cunningham and Peter Song and Ross Brimberry, who were also involved in the project. And in particular, I want to mention Rudy Tanzi and Dmitry Prokopenko at MGH, who did the initial identification of ACE1R1279Q. I also want to thank my funding sources, in particular the Cure Alzheimer's Fund, which funded this work.